Imagine a quiet invasion occurring right within your stomach. A tiny bacterium named Helicobacter pylori stealthily moving in. While small and seemingly harmless, this bacterium has a dark side. In fact, it is officially recognized by the World Health Organization as a Class 1 carcinogen, meaning it's proven to directly cause cancer. But how could such a small creature cause something as severe as cancer? Helicobacter pylori, or H. pylori for short, starts its journey by settling in the lining of your stomach. Here, it cleverly releases a harmful protein called CAGA, a microscopic saboteur that enters the cells lining your stomach walls. Once inside, CAGA wreaks havoc. It disrupts the way your cells grow, divide, and behave, like a criminal mastermind causing chaos within an orderly city. When CAGA enters these gastric cells, it doesn't stop there, it goes even deeper. Imagine flipping multiple switches in a control room, causing systems to malfunction and sending wrong signals everywhere. CAGA does just that by activating various cellular pathways that shouldn't be constantly switched on, such as B-catenin, PI3K/AKT, GSK3B, and STAT3. These pathways normally regulate critical functions like cell growth and survival, but when hijacked by CAGA, they promote uncontrolled growth, inflammation, and even mutations. The perfect recipe for cancer development. The chaos doesn't end there. When CAGA meets certain enzymes like SRC family kinases, it's modified through a process called phosphorylation. This modification turns CAGA into an even more potent villain, enabling it to interact with a protein known as SHP2. This interaction amplifies certain signals, causing gastric cells to lose their original shape, becoming elongated and migratory, a shape described vividly as the hummingbird phenotype, making the cells more invasive, another step toward cancer. Even unmodified CAGA can cause trouble by messing with a crucial protein complex called E-catenin B-catenin. Normally this complex acts like glue, holding cells together tightly. But CAGA disrupts this bond, causing B-catenin to accumulate inside the cell nucleus, where it activates genes associated with cancerous growth. Essentially, the cell is tricked into behaving like it's permanently switched on, relentlessly dividing and surviving in an environment that would usually halt its growth. Beyond CAGA, H. pylori employs another weapon known as VACA, vacuolating toxin A. VACA makes cells sick by forming channels in their membranes, triggering cell death, inflammation, and further promoting cancer. Like CAGA, VACA can also activate the B-catenin pathway, adding yet another layer to this bacterium's harmful strategy. Recent studies have revealed even more cunning tricks by H. pylori. It uses tiny particles called membrane vesicles to deliver harmful compounds into cells, directly manipulating their life and death decisions. In short, Helicobacter pylori quietly but effectively manipulates cells at multiple levels, from their surface to their deepest genetic controls, gradually pushing healthy gastric tissue toward cancer. Scientists continue to unravel this bacterium's complex schemes, hoping that by understanding its strategies, they can find effective ways to stop its destructive path and protect millions from stomach cancer.